Once the SD card from the LCpro T is inserted into a card reader in a laptop or a PC, the data files can be opened as here. At the top are several bitmap images with the name of the graphical plots that were previously saved. If I open the latest plot, this shows all data points plotted in the sequence file when an ACI curve was plotted. This Excel spreadsheet formatted file is the result of pressing Export when plotting a graph, swiping down to reveal those extra options. Here we have 3 columns denoting the time that the graph was begun and the number of data points plotted. The second column shows the parameter that was plotted on the y-axis and all of the corresponding values. In the third column is the parameter plotted on the x-axis and values. So here we have A against CI, rate of photosynthesis, denoted by A, against intercellular CO2, or CI. Here we have opened the main data log file, which was named under the logging menu. At the top is the number of data points or records. This line gives information as to the system, the LCpro T, and its 5-digit serial number. In the first column are all of the data points or records, followed by date, time, and then we go on to 3 parameters that begin with E. This denotes water vapour. In column D we have E-ref, E-reference value. Here we were controlling E at ambient, which you can see in the third column. The values are given in millibars, so here 18.2 millibars to 20 millibars. Column E denotes delta, or the change, the difference in E value. And from this value can be worked out the E analysis from the leaf chamber. So this value is telling you the difference between the E reference and the E analysis as given in the leaf chamber. So this is the change, the difference seen in water vapour created by the leaf within the chamber. E control shows that in microclimate control we were controlling its ambient. In column G, C reference denotes the C reference tells us the achieved values from within the analyzer that are given from the CO2 cartridge installed. These values are given in parts per million. These values were created through the sequence file and programmed in to decrease and increase and then decrease again to the start value. In column H, delta C denotes the difference between the C reference value and the C analysis value from the leaf chamber. C control is the user set absolute values that were set through the sequence file. And here we can see the difference between the achieved value and the set value in the order of 2 to 3 ppm. The Q leaf value refers to the light or PAR reaching a leaf, and here it is 435 micromole per meter squared per second. Q mode is the source of the light reading given through the climate control. Q white is not applicable in this case because an RGB light unit was fitted. Q red is the red LED output in micromoles per meter squared per second. Q green, the green LED output, and the blue LED output. Here you can see the relative quantities in actual values. TCH is temperature in the leaf chamber in degrees centigrade. Temperature control denotes the user set value in the climate menu, in this case 25 degrees centigrade. T leaf denotes the leaf temperature as given through the springy sensor in the lower jaw. And here we can see the method of calculating leaf temperature. In this case, calculated through the sensor. The other option here would be measured, which would be using the manual placement sensor that can be connected to the lower jaw. In column T is U, or the airflow. This is the achieved value, and next to it the user set airflow value, in this case 200 micromoles per second. P is atmospheric pressure in millibars. CI, a key parameter to look at for stability values when starting an experiment. This is the intercellular CO2 in VPM or ppm. E is the rate of transpiration in millimole per meter square per second. GS is stomatal conductance, again in millimole per meter squared per second. A, rate of photosynthesis in micromole per meter squared per second. These values were used in the graphical plot along with the CI values. Area denotes the leaf area or area of the chamber window. This is a default value set to the type of chamber being used. There is also the option to change the area manually if, for example, a leaf in the chamber does not quite cover the chamber window area. RB is boundary layer resistance in meters squared per second per mole. The next four columns refer to the GPS module fitted in the LCpro T. In this case, measurements were taken indoors and no values are shown here. When outdoors, there would be a value for each and a fix indicator to give the strength of the GPS signal. The final column is the chamber configuration. In this case, a broadleaf chamber was being used.